Hi, everyone, and welcome to our second lecture, which will cover the first part of Chapter 2, which is called The Chemistry of Life. Chapter 2 is an important chapter because it really lays the foundation for a lot of other topics that we are going to be discussing later in the semester. So that's why we're going to take it slow, divide it up across 2 weeks, and really take our time exploring the chemistry of life. And before we get started, let's talk about the elephant in the room, which is that word chemistry. Because you may be thinking, what is that word doing here? I signed up for a biology class. I'm not interested in chemistry. If I wanted to learn chemistry, I would be in a chemistry class. But the fact of the matter is that biology and chemistry are closely intertwined with each other, especially in modern times. And you really can't understand a lot of the most important concepts in biology without at least having some foundation in chemistry. And that's because modern biology is so advanced that we understand how living organisms function on a very deep, complex, fundamental level that gets all the way down to the workings of the atoms and molecules that make them up. And early biology was not like this. Even just a couple hundred years ago, biology took a much more superficial look at living organisms. So, asking questions about what types of organisms there were, what their body structures were, what habitats they lived in, etc. But today, biologists really dig deep when they are looking at the way that organisms function. And because of this, we are able to come up with solutions to problems in biology. And this level of exploration and problem solving has been made possible by tools and technologies that have vastly expanded the capabilities of the biologist as a scientist. Everything from the modern microscope to genetic sequencing technology, the ability to genetically engineer cells, the ability to grow or raise organisms in a laboratory environment, or even just grow parts of organisms in petri dishes, like that second to left photo where there's a sheet of skin that's being grown in a dish for a skin graft onto a burn patient. To give you a better picture of how chemistry is intertwined with biology and why we need to understand chemistry to solve problems in biology, I've come up with a few examples of questions that modern biologists can and have asked. For example, what genetic mutations make a person more susceptible to colon cancer? Answering this question requires knowledge of chemistry because genetic mutations occur in DNA. And as we will learn, DNA is one of the major chemicals that makes up living organisms. Mutations are changes in the chemical structure of DNA. So to understand and identify these mutations, we would have to know something about the chemistry of DNA. How can we modify the chemical structure of the antibiotic vancomycin to make it more effective against antibiotic-resistant bacteria? So antibiotics are the drugs that we use to treat bacterial infections, and all drugs, all medications are chemicals. And one growing problem with antibiotics is that bacteria are becoming resistant to them. They no longer work. So scientists are constantly searching for new antibiotic chemicals and looking for ways to change the chemistry of our existing antibiotic medications to try to outsmart the resistant bacteria. How much magnesium should an elite athlete consume daily during training? So, like all minerals and vitamins that we get in our diet, magnesium is an important chemical that our bodies need in order to function. And figuring out the optimal level that a person should consume and what form they should take it in is a question that involves both biology and chemistry together. What concentration of phosphate fertilizer can be applied to crops without the runoff causing toxic algal blooms? This is actually a very real problem that farmers face. Phosphate, which is, again, a chemical, is essential for crop plants. But if too much of it leaches into surrounding bodies of water around the cropland, it can cause an explosion in the growth of toxic algae that can and has made people very sick and killed a lot of wildlife. So, how do we find the right balance between providing enough phosphate for the crops without overdoing it and causing toxic algal blooms? So, hopefully, this smattering of questions gives you an idea of how important chemistry is in biology and why we have to learn a little bit of chemistry to lay the foundation for the rest of our journey in this class. But instead of just telling you, I also want to tap into your curiosity as well in our first checkpoint question. I'd like you to add to this list of questions by thinking of your own original question about a biology topic that would incorporate or rely on chemistry principles to find the answer. So now that I've given you an explanation of why we bother learning about chemistry in a biology class, let's take a look at what we're actually going to be learning. As I said, this chapter is going to be spread out over 2 lectures spanning 2 weeks. And in the first part, this lecture recording, we are going to answer a few questions. And those questions are, what are atoms, ions, isotopes, and molecules? Why is water such an important molecule for living organisms? And what are acids and bases? And then, once we've laid those foundations, in part 2, we will take a look at what are living things made of on a chemical level. So, starting with part 1, this lecture, let's jump right into our first question, which is, what are atoms? Atoms are the basic unit of matter that everything is composed of, including both living things and non-living things. Any object that exists, if you break it down into its smallest, ultra-tiny pieces, those pieces are atoms. As we will see in a moment, there are many different types of atoms, but they all share a basic structure. They have a central cluster of particles called the nucleus, and then one or more rings around the outside called orbitals that contain these little orbiting particles. You've probably seen a diagram of the solar system before with the planets revolving around the sun. That's very similar to the structure of an atom. And what you see on this slide is, of course, just an illustration. It's just a drawing. But in 2013, the first ever image of an atom was captured using an extremely powerful microscope. And this had never been done before this point, but in this image, we can see the structure laid out with that central cluster of particles and then the rings of orbiting particles. So what are those particles that are found within the atom? Well, we call them subatomic particles, and there are 3 of them. There's the protons, which are positively charged and located in the nucleus. Then the neutrons, which have no electric charge, they're neutral, and they're also found in the nucleus alongside the protons. And then lastly, the electrons, which have a negative electric charge, and these are the ones that are orbiting in the orbitals. And the location and charge are not the only features that distinguish these particles from each other. Mass is another feature that differs between them. So protons and neutrons both weigh 1 Dalton each. And what is a Dalton, you ask? Well, it is a very, 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 very tiny unit of measuring weight or mass. If we were to express the mass of a proton or neutron in pounds, pounds being a unit that we're more familiar with, then 1 Dalton would be 0.0000, that's 26 zeros, and then a 3 pounds. So unimaginably tiny. But amazingly, even though protons and neutrons each weigh only 1 Dalton, electrons weigh so little compared to even a proton or a neutron that their mass is not even counted towards the mass of the atom. So the mass of any given atom in Daltons is simply the number of protons plus the number of neutrons, because the electrons are so small they don't count. So to summarize, protons are found in the nucleus. They have a mass of 1 Dalton and a positive electrical charge. Neutrons are also found in the nucleus, and they have a mass of 1 Dalton but no electrical charge. And electrons are found in the orbitals. They have no mass, and they have a negative electrical charge. So in this checkpoint, I would like you to take a look at this carbon atom on the right-hand side of the slide and tell me, what is the mass of this atom in Daltons? So now that we know what an atom is, let's talk about the different types of atoms. The different types of atoms are called elements. An element is a type of atom with a specific number of protons in its nucleus. For example, atoms of the element carbon have 6 protons in their nucleus. If we're talking about the element nitrogen, atoms of nitrogen have 7 protons in their nucleus. If we're talking about oxygen atoms, they have 8 protons in their nucleus. So this number of protons is what makes an element an element. Carbon is, by definition, an atom with 6 protons in its nucleus, and that never changes. Those 6 protons are what make carbon carbon. And all of the different elements that are known to exist in the universe are organized into a chart that you may have seen before. It's called the periodic table of elements. So each of the boxes in this chart represents one single element. There's a box for carbon, there's a box for nitrogen, etc. In total, there are well over 100 elements on the periodic table, but only 92 are naturally occurring. There are others, but they have to be artificially created in a lab. And then out of those 92 naturally occurring elements, 25 of them are responsible for making up the structure of living organisms. And those are the ones that you see highlighted in some sort of color here in this periodic table, either orange, green, or pink. And then out of those 25, just 4 of them are responsible for making up 96.3% of the mass of living things. Those are the ones that are specifically highlighted in orange: hydrogen, carbon, nitrogen, and oxygen. So we're going to see these 4 come up repeatedly, especially as we get into part 2 of this lecture and we start talking about what living things are made of on a chemical level. Now let's zoom in and take a look at one of these squares on the periodic table more closely. And as I said, each square represents one element, and this particular square is showing carbon. Every element has a 1 or 2 letter abbreviation, which is called its elemental symbol. And for carbon, the elemental symbol is C. There are also a few numbers on here. Up top is something called the atomic number, and this is a whole number, meaning it has no decimal, that represents the number of protons in atoms of this element. So carbon has 6 protons. That never changes. That's what makes carbon carbon. And then down below the elemental symbol, there's another number, and that's called the atomic weight. So we saw before that the weight or mass of an atom comes from its protons and neutrons. So that's what this is. It is the average number of protons and neutrons in the carbon on Earth. Now you may notice that this number, the mass, the weight, is not a whole number. It's not just 12, it's 12.01. So where did that 0.01 come from? If protons and neutrons each weigh exactly 1 Dalton, how can we get these fractions of a Dalton in there? The answer is that not all atoms of a given element have the same number of neutrons. They'll always have the same number of protons, but not always the same number of neutrons. And this number that you see on the periodic table, that is the average mass. The reality is that most carbon in the world has 6 protons and 6 neutrons, giving it a mass of 12, and we call that carbon-12. But a very small amount has 6 protons and 7 neutrons, or even 8 neutrons, which gives it a mass of 13 or 14, which we call carbon-13 and carbon-14. These are all different isotopes of carbon. Isotopes are atoms of the same element that have different masses because they have different numbers of neutrons. So the reason why the mass on the periodic table is not a whole number is because it is an average. Almost all carbon has a mass of 12, but a very small amount has a mass of 13 or 14, which nudges the average just slightly up above 12. So we've been using carbon as an example, but every element has isotopes. Here's another example. Nitrogen, which is another one of those 4 very common elements found in living organisms. All nitrogen atoms have 7 protons, and 99.6% of nitrogen atoms have 7 neutrons, which gives it a mass of 14. But then a small fraction of nitrogen atoms have 8 neutrons instead of 7, which gives it a mass of 15, and then a completely negligible amount has just 6 neutrons. So overall, the average mass of nitrogen is very close to 14, because almost all nitrogen on Earth weighs 14, but it's just barely above 14 because there's that contribution from the isotope that weighs 15 instead of 14. So now we know what isotopes are, but why should we care? What uses do isotopes have in the world of biology? I will give you a few examples of how isotopes are useful in biology. So there's a medical technology called a PET scan that captures information about things like your blood flow and oxygen levels in the body by tracking certain isotopes in the body. So if a person is experiencing symptoms that are consistent with failure of a particular organ, a PET scan can confirm this. Another example, looking at different levels of isotopes in fossils can also be used to determine the date when the fossilized organism died. And this is how we can learn about organisms of the past, like dinosaurs. In addition, certain radioactive isotopes that are created in the lab emit radiation, and these isotopes are routinely used in chemotherapy for cancer as well as in other disease treatments. Radioactive isotopes are also used in a process called irradiation, which is where radiation from radioactive isotopes is used to kill bacteria in meat and fruits and vegetables, which could contaminate them and cause spoilage. And so these food products will be irradiated before they're shipped to a grocery store to ensure that they maintain their shelf life and that they're safe to eat. So these are a few ways that isotopes are useful in the sphere of biology. And this brings me to a set of 2 checkpoints that will build off of each other. In this first checkpoint, I would like you to tell me, based on this image, how many protons are in the nucleus of an iodine atom. And in this checkpoint, we're going to continue to focus on iodine. The most common isotope of stable, naturally occurring iodine is iodine 127, meaning iodine with a mass of 127. However, iodine 131 is a separate radioactive isotope that scientists can make in a lab, and the reason why they do this is because iodine 131 is used in chemotherapy to treat thyroid cancer. This type of chemotherapy is called radioactive iodine therapy, and in it, iodine 131 is administered to a patient through an IV or a pill, and then it travels to the thyroid gland and emits radiation and destroys the cancerous thyroid cells. So based on this information and your answer to checkpoint 3, how many neutrons are in the stable, naturally occurring iodine isotope, and how many are in the isotope that's used in radioactive iodine therapy? And just as a hint and reminder, you can't answer this question without relying on the answer that you came up with to the previous checkpoint 3. So we've established that protons are the subatomic particles that define an element's identity. So the protons always stay the same. And we've also established that neutrons can vary to create different isotopes of an element. But what about the electrons? Electrons are the third subatomic particle found in atoms alongside the protons and neutrons. And electrons in an atom can vary too. Atoms normally have the same number of protons and electrons, making them neutral. So in other words, here we have an atom of the element fluorine. Fluorine has 9 protons, which means 9 little positive charges in the nucleus. And a fluorine atom also has 9 electrons, which means 9 little negative charges in the orbitals. So if you take the atom as a whole, the 9 positive protons and the 9 negative electrons cancel out. So the atom is neutral. But what happens if that electron number changes? If and when the atom gains or loses an electron, it becomes an ion. An ion is a particle with an electric charge due to having gained or lost one or more electrons. For example, if this fluorine atom gains one electron, it goes from having 9 electrons to 10 electrons. The number of protons is still 9 because it's still fluorine. So now this fluorine ion has a total electric charge of -1. It has one more electron than it has protons. Now again, we find ourselves in a position where we know what ions are, but why should we care? What's the relevance of ions in biology? So like we did with isotopes, I'll give you a few examples. So firstly, the signals in our nervous system travel through our body using the electrical charges carried by ions. Every time you have a thought, every time you move a muscle, you owe ions for carrying the transmission of that activity through your brain and your nervous system. Ions are also used as food preservatives because they can dehydrate and kill bacteria that would spoil food. Another place where ions are relevant to our lives is that ions are pretty much synonymous with electrolytes. And you can sort of see this in the name. Electrolytes are electrically charged particles, which is the definition of ions. And electrolytes are very important to us to maintain our hydration as well as pH levels in the body. And that's why if you're dehydrated, doctors recommend drinking something like Pedialyte, which is rich in electrolytes. And if we look at the nutrition label for Pedialyte, we can see that the ions that are found in this beverage include magnesium, potassium, selenium, zinc, and chloride. All of these are elements that you find on the periodic table. And in the form of Pedialyte, they've lost or gained electrons and they're now ions. So let's take a look at one of these ions a little bit closer in a checkpoint. Let's pull out magnesium and take a look at that. So magnesium ions have an electrical charge of +2. Knowing this, how many electrons are there in a magnesium ion? And to give you a hint, your first step here should be to figure out whether this charge of +2 means that the magnesium ions have lost or gained electrons. Does a charge of +2 mean that electrons were added or that they went away? And then figure out the number of protons found in magnesium from this periodic table square. And then lastly, keeping in mind that the number of protons does not change, figure out how many electrons there must be compared to the number of protons. So we know that ions are atoms that have gained or lost one or more electrons. But that begs a question of where do ions get those extra electrons from? And when they lose them, where do they go? And the answer is the same in both cases. They are gained from and lost to other atoms. In other words, ions are always coupled together. When one atom loses an electron and becomes positively charged, that's because it gave away its electron to another atom, which then becomes negatively charged. And that's what you see happening in this animation right here, which shows an electron transferring from an atom of sodium on the left, which is shown with the elemental symbol Na, and it has the blue electrons, to the atom of fluorine on the right, which is F with the red electrons. As a result, the two ions that are created here carry opposite charges. The sodium becomes positive and the fluorine becomes negative. And now they are attracted to each other, much like the positive and negative poles of magnets are drawn towards each other. These ions are drawn towards each other as well. What you've just observed in this animation is the formation of an ionic bond, a bond formed when one atom takes electrons from another atom. And then they gain opposite charges, and then they have this linked attraction to each other. And as a result, a molecule is formed. A molecule is a group of atoms held together in stable association. So in this case, we have a molecule of sodium fluoride consisting of one sodium ion and one fluorine ion held together by an ionic bond. Now, why does this phenomenon happen? Why do atoms start moving their electrons around in the first place? Well, it's because Atoms and ions are most stable when their outermost orbital has exactly 8 electrons in it. So at the beginning of this animation, this is not the case for either sodium or fluorine, as you can see here. Sodium on the left has just 1 electron in its outer orbital, and fluorine on the right has 7. But if sodium gives that 1 electron to fluorine, now fluorine has 8, and sodium has gotten rid of its outer orbital entirely, revealing an orbital underneath that does have 8. So both sodium and fluorine are happy and satisfied with this transfer happening. Now, transferring electrons is not the only way that atoms can achieve 8 electrons in their outer orbital. There's another type of bond that can occur between atoms called a covalent bond. And this is where, instead of transferring electrons to form a molecule, atoms share electrons with each other. Any electrons that are shared will count towards the outer orbital of both of the atoms involved in the arrangement. So in this example, we have 2 atoms of chlorine. Cl represents chlorine. If we take a look at the outermost ring, the outermost orbital on chlorine, we can see that chlorine atoms have 1, 2, 3, 4, 5, 6, 7 electrons in their outer orbital. So in this state, they're not satisfied. But if these 2 atoms come together and they each bring just 1 of their outermost electrons to the table, then they can form a covalent bond where this pair of electrons that they have created counts twice. It counts towards the orbital of the chlorine on the left, and it counts towards the orbital of the chlorine on the right. So by sharing these 2 electrons with each other, they come into this stable molecule arrangement. So we've established that there are 2 types of bonds: ionic bonds, where electrons are transferred and create electrically charged ions that are attracted to each other, and then covalent bonds, where 2 atoms each bring electrons to the table and they share them with each other so that they count towards both orbitals. But how do we know which type of bond will happen in any given situation? Well, this has to do with the identity of the elements involved. So what you're seeing here is a periodic table that has been color-coded based on whether each element is a metal, a non-metal, or a metalloid, which a metalloid is a substance that has some qualities of metals and some qualities of non-metals. But we just want to focus on the metals, which are shown in silver, and the non-metals, which are shown in bronze. Some examples of elements that you can see in the metal section are aluminum, zinc, iron, cobalt, nickel. Some examples of nonmetals are hydrogen, carbon, nitrogen, oxygen. Often just from our experiences in the world, we can tell from the name of the thing if it's a metal or a nonmetal. Now here's the deal. Nonmetal elements are quite electron greedy. Because of this quality, they will take electrons from atoms that are willing to give them up. The elements that are willing to give them up are the metals, because metals are not electron greedy, and they readily give their electrons away. So when a nonmetal forms a bond with a metal, it will be an ionic bond because a transfer of electrons occurs. On the other hand, when a nonmetal element forms a bond with another nonmetal element, they are forced to share with each other because they are both greedy and both want electrons, so neither one is willing to fully give up their electrons. So nonmetals form covalent bonds with other nonmetals. And why is this important for biology? I promise we are getting there. We just have to go a little further before the significance becomes clear. So we said that nonmetals are more greedy than metals. However, not all nonmetal elements are equally greedy. And when they get together in a covalent bond with another nonmetal, the relative level of greediness between the 2 elements in the bond also affects its qualities. So when you have a bond between 2 nonmetals, that are equally as greedy as each other. We call this a nonpolar covalent bond. A nonpolar covalent bond is formed when two nonmetal elements share their electrons equally. This happens when a bond forms between two nonmetals that have a very similar level of electron greediness. And a great example is the one we just looked at. Two chlorine atoms coming together will form a nonpolar covalent bond. Obviously, because they are the exact same element, they have the exact same level of electron greediness. So they end up sharing those electrons equally because they're each tugging on them with the same level of force. Now let's take a look at what it looks like when two nonmetals get together and share their electrons in a covalent bond, but they're not sharing them equally. We call this type of bond a polar covalent bond. So the electrons are still being shared, but they're not shared equally. A polar covalent bond is formed when two nonmetal atoms share their electrons unequally. This happens when a bond forms between two elements with different levels of electron greediness. So one of the elements involved in the bond is greedier than the other. An example of two elements that would engage in a polar covalent bond are hydrogen and chlorine. So hydrogen is a bit of an exception to the rule, where instead of wanting 8 electrons in its outer orbital, hydrogen only wants 2 because it's so small, there's actually not enough room for 8. But despite that, both elements here can get what they want if they each bring an electron to the table and form a covalent bond where they're sharing this pair of electrons with each other. However, it turns out that chlorine is more greedy for electrons than hydrogen is. There's a disparity there. So they're sharing, but when they enter this arrangement, they will share the electrons unequally. Chlorine will pull them closer to itself and away from the hydrogen atom. And as a result, the right side of this molecule, the one around chlorine, is now slightly negative because it has more of the negatively charged electrons clustered in that area, while the left side is slightly positive because the electrons have been drawn away from that area, which leaves the positively charged protons to be the dominant force in that area. And that's why this is called a polar bond. When we think about the word polar, what it means is that there's two opposite ends. The North Pole and the South Pole, these are the polar opposite ends of the Earth. So in this polar bond, there is a positive end and a negative end, and that's why we call it polar. So to summarize, we have 3 types of bonds that are formed between atoms when they become molecules. In ionic bonds, electrons are lost from one atom and gained by another. And these types of bonds occur between an element that is a metal, which gives up its electron, and a nonmetal, which receives it. Covalent bonds involve two nonmetals sharing their electrons, and there are 2 types. In a nonpolar covalent bond, the elements have similar levels of electron greediness, so the electrons are shared equally because they're both pulling on those electrons equally. And then lastly, in polar covalent bonds, there's a difference between the electron greediness of one of the elements and the other. One pulls harder on the electron, so you end up with an unequal distribution, and one end is positively charged and the other is negatively charged. So this brings us to another checkpoint. And in this checkpoint, I'd like you to look at each of these 3 molecules, potassium chloride, carbon dioxide, and methane, and classify each one of them as having ionic or covalent bonds taking place. And there's no need to state whether the covalent bonds are polar or nonpolar, just whether they are ionic or covalent. And it may be helpful for you to see that version of the periodic table with metals and nonmetals distinguished from each other. So I will flip to that slide in just a moment so that you can use it to answer this question. So now we finally arrived at the main reason why we are suffering through all of this stuff about molecules and bonds and polar and nonpolar. Now let's talk about water, H2O. Water is an extremely important molecule in biology. Water is the basis for all living organisms, whether you're a plant or an animal or a fungus or a bacteria. Water is found inside of cells. It is found outside of cells. The weight of the average human body is about 55 to 65% water. But why? What makes water so important? Water actually has multiple special qualities that are brought about by the fact that it is a polar covalent molecule. It's made of 2 hydrogen atoms and 1 oxygen atom. And the hydrogen atoms each share their electron with the oxygen atom, but the oxygen atom is more electron greedy than the hydrogens. So it draws those electrons toward itself, and you end up with a polar molecule. The hydrogen ends are each slightly positive because they have a lower concentration of electrons, and the oxygen is slightly negative because it has more electrons. And the polar nature of the water molecule and the fact that it has these negative and positive ends gives it a lot of the properties that are necessary to its ability to support life. Because when you're looking at a body of water or water inside of your cells or inside of an organism, you're not just talking about one water molecule. You're talking about countless water molecules together in a collection. And when you have a collection of water molecules like this, the positive end of one molecule is attracted to the negative end of another molecule. These yellow dotted lines that I've added represent the network of attractions between separate water molecules. And you can imagine how the water molecules sort of stick to each other and remain as a cohesive unit. And this arises from the fact that they are polar molecules because, of course, if they didn't have these positive and negative ends, the attractions between them would not exist. So what are the consequences of the fact that water is a polar molecule with this network of attractions? We're going to discuss 3 properties that make water specially able to support life. The first of which is that water molecules have cohesion and adhesion. Cohesion describes the fact that water molecules stick to each other, and adhesion is the fact that water molecules will stick to other stuff too, so long as that other thing is also a polar molecule. And cohesion is something you've probably observed before. If you've ever done that little experiment where you drop water onto the surface of a penny and you watch this big dome form and you're expecting it to burst and flow off the penny, but it just keeps getting taller, the reason why water is able to support this shape without collapsing off the penny is because of cohesion. The water molecules stick to each other. If you do this with another liquid like ethanol, for example, you can't achieve this dome shape because the cohesion just isn't there. In nature, these cohesive and adhesive forces allow water to be transported from place to place. For example, water is pulled through plant roots and distributed throughout the plant because of its cohesion and adhesion. In this demonstration here, we can see a paper towel being bent and then dipped into a cup of colored water on the right, and then there's an empty cup on the left. And the water gets drawn up into the material of the paper towel, and then the force of gravity pulls it down into the empty cup. The reason why water is pulled up into the paper towel is because of the adhesion of the water molecules, the attraction of the water to the paper towel, and then cohesion pulls other water molecules along behind them. And this is exactly how water moves through plants as well as through other living organisms. So without cohesion and adhesion, this transport of water would not occur. Another important property that arises from the polar nature of water is water's ability to maintain a stable temperature. So water has this remarkable ability to keep a stable temperature even when it's absorbing a lot of heat. A thought experiment that I like to pose as a demonstration of this point is to ask you guys to imagine that it is a hot summer day in southern Arizona. It's 115 degrees outside. You've got your car parked out front and there's a swimming pool in the backyard. Which would you rather do? Would you rather go lay on the hood of your car in a swimsuit, or would you rather jump into your swimming pool in the swimsuit? So obviously we all choose the pool because the metal hood of the car is blazing hot, but the water in the pool will still be a nice, pleasant, tolerable temperature. And that's not because one of these things is getting more heat than the other. The weather conditions aren't any different in the front yard versus the backyard. It's because of the material that they're made out of and each of those materials' abilities to stabilize temperature. So the temperature of a substance is on a physical level related to the motion of its molecules. The faster and more erratically those molecules are moving, the higher the temperature of the substance. When it comes to water, those attractions that we pointed out between individual water molecules keeps the motion of the molecules calm and steady. They can't move erratically as easily because they're stuck together in this network of attractions, which is what this animation is showing you. So the temperature of the water in the pool remains more stable, unlike the temperature of the metal in the hood of the car, because the molecular motion is kept subdued by the polar attractions between water molecules. Now in this checkpoint, what I would like you to do is brainstorm on what would be some consequences for living organisms if water behaved more like metal in terms of its temperature stability. So if water wasn't made of polar covalent bonds and it had these attractions that keep its temperature stable, what would that mean for living organisms? And you can talk about what it would mean for humans, what it would mean for plants, land animals, aquatic animals, whatever type of living thing you want to explore in this question. This brings us to the third and final property of water that comes from its polar nature. And this is that ionic molecules and polar molecules are easily able to dissolve in water. And that's because if we can return to this picture, those attractions between water molecules can be substituted for attractions to other molecules that themselves have full or partial electric charges. For example, if we drop a molecule of salt, sodium chloride, into a sample of water, it fits right in because the sodium ion has a positive charge, so it's attracted to the negatively charged ends of the water molecule. And then the chlorine atom has a negative charge, and that's attracted to the positively charged ends of the water molecules. So it fits beautifully into this network of attractions. So water can dissolve polar molecules and ionic molecules, and this is essential for living organisms because living things are full of water, and many of the important biological molecules that support our functions, like for example salts and sugars, need to be able to dissolve in water so that they can be transported, they can be involved in chemical reactions. This short video clip shows a sample of salt, which is an important molecule that living things rely on, dissolving into a glass of water. On the other hand, while ionic and polar molecules dissolve well in water, nonpolar molecules do not dissolve in water at all. So some examples of nonpolar molecules you're probably familiar with are oils and fats. And if you've ever seen oil poured into water, then you've probably observed this phenomenon where the oil will not mix with the water and it will instead separate itself into a different layer. And this is because nonpolar molecules, like fats and oils, lack any sort of attraction to other molecules. If you drop a nonpolar molecule into a sample of water, and here again we're using the example of two chlorine atoms bonded together as our nonpolar molecule, this nonpolar molecule does not fit within this network of attractions. It is not attracted to the water molecules, and the water molecules aren't attracted to it. And so it ends up getting pushed into a separate layer. And this is why oil behaves the way it does when it's combined with water. So not only does the polar nature of water make it very effective at dissolving certain things, namely polar and ionic things, it also makes it so that water wants nothing to do with nonpolar things. And this property is going to be an important feature that explains how and why cells form in nature, but we're going to get to that in a future chapter. For now, the only remaining topic that we have left to tackle in this lecture are acids and bases. So you may already know from your previous learning that acids and bases are substances on opposite ends of the pH scale. But what does the pH scale actually tell us? Well, the pH scale measures the concentration of hydrogen ions in a particular substance. Whether something is an acid or a base, and how acidic or basic it is on the scale is dictated by the concentration of hydrogen ions that can be found in that substance. And when we say hydrogen ion, what do we actually mean? Well, remember that an ion is an atom that has an electrical charge resulting from having gained or lost an electron. In the case of the element hydrogen, hydrogen is the smallest and simplest element in existence. You see from its periodic table square that it has an atomic number of 1. So this means that a hydrogen atom has 1 proton in its nucleus and 1 electron in its orbital. An atom of hydrogen becomes an ion when it loses that electron by giving it away to another atom. And when this happens, all that's left behind is the proton in the nucleus. So let's put together what we've learned about atoms and ions in this lecture to the test again in another checkpoint. What is the mass of a hydrogen ion? And what is the electric charge of a hydrogen ion? So now that we know what the pH scale measures, which is the concentration of hydrogen ions, getting back to the scale itself, the scale runs from 0 to 14. Right in the very middle of the scale is the number 7, which is the value assigned to pure water. Pure water is considered neutral. It's neither acidic nor basic. As you go lower and lower on the pH scale towards the number 1, the concentration of hydrogen ions actually increases and things get more acidic. And conversely, if you go higher and higher on the scale, the concentration of hydrogen ions decreases and the substance becomes more basic. So anything below 7 is an acid, and the lower the number, the more hydrogen ions, the stronger the acid. We can see something like battery acid is very strong around pH of 0. So if we go a little higher, we have stomach acid at 1, lemons, soda, tomatoes, coffee, and milk is just slightly acidic. Anything above 7 is basic, and as you go higher on the pH scale, the value indicates that there are fewer hydrogens and the base becomes stronger. So some mildly basic substances that you might know are human blood, egg whites, and then farther up the scale, you get to really strong bases like bleach or drain cleaner. Now, what significance do acids and bases have to living organisms? So typically, cells have a pH range in which they function best. For human cells, that range is right around a neutral pH, pH 7.2 to 7.6. Outside of that range, human cells incur severe damage, and so it's very important that cells are able to maintain the range that is best for them. However, you may have noticed that a lot of the things that we put into our bodies are not neutral in their pH. Lemons, soda, tomatoes, coffee, milk, all of these are on the acidic end of the scale. So how can we consume these things and put them in our body without endangering our cells by throwing the pH out of whack? The answer is that our bodies contain chemicals called buffers that allow us to do this. Buffers are chemicals that absorb or release hydrogen ions in response to pH changes, and this allows the cell's pH to stay in that safe range. And this is part of homeostasis, which you may remember learning about in Chapter 1 as being one of the characteristics shared by all living organisms, the ability to maintain stable internal conditions. One example that pertains to humans is our blood. Our blood contains buffering chemicals that keep the pH in the safe range, even when we put things in our bodies that might threaten to disrupt the pH. Now, we've been talking about the human body and its preference for a pH that is approximately neutral, but not all living things prefer a neutral pH environment. So for example, the microorganism Sulfolobus prefers acidic pH environments and thrives in the acidic sulfur pools found in Yellowstone National Park, which you see here in this picture on the left. And those pools have a pH of about 3. On the opposite end of the spectrum, the microorganism Natronobacterium thrives in Lake Magadi in Kenya, which is seen here on the right, and that has a very basic pH of about 11. So in this final checkpoint of the lecture, I'd like you to tell me which of these two environments, Yellowstone National Park sulfur pools or Lake Magadi, has a higher concentration of hydrogen ions. Is it the one with the pH 3 or the pH 11? And then once you finish this checkpoint, you have come to the end of this chapter, and I'll see you in the next lecture.